in an extra topic which is not related to a time series, but it's just to let you know that a lot of the methods that we will present for time series analysis can also be used to analyze images. And that's why I'm presenting this topic here, which is not related to time series analysis, but we will use networks. So basically, the first part I want to explain is what techniques can we use to uncover similarities between lasers and neurons, and why do we care? And then we will analyze other methods to identify transitions. I will put examples of EEG signals and vegetation fields, but the techniques are quite generic and can be used for analyzing other type of dynamical behaviors in which there is some sort of change, tipping point, something that changes the behavior. You can detect a change in behavior. Okay. So yeah, these are two time series. One is experimental time series detected in a lab with an oscilloscope, and this is the intensity emitted by a laser, and you can see some pulses. And these pulses have apparently similarities with spikes. Those are spikes simulated with a neural model. And we are interested about trying to see if they are really similar or just look similar. The interesting thing is that the timescales are very different, are orders of magnitude different. And in the end, the goal is, can we use lasers to implement neurons? And neurons that actually act genuinely as biological neurons. And this, just to give you some physics of this, I'm not going to talk about the experiment, but it's a very simple experiment. It's one that semiconductor laser. This is a very cheap, it's the most popular type of laser used in sensors, in optical communications. These are very cheap technology because it's compatible with electronic chips. And that is the Ficus-Nagumo model, which is also a very well-known model for representing a neuron. And Anna, I think Anna mentioned it, yeah. Okay, the difference, important difference between them is the fact that here we have some delay because the light goes and comes back. This is the experiment with a mirror. And this means that the system has memory. This system is low dimensional. It has two variables that describe the dynamics of the system. Okay, so the method that we're going to use to see if the time series are really some significant similarities or not is known as ordinal analysis. Many of you are familiar with this method. Some of you are not, so I will just briefly introduce this without going too much in details. Suppose you have three numbers, for example, 2, 5, and 7. The way they can be organized, they can occur, the temporal order can be like a trend, positive trend. There can be some oscillatory behavior, or they can be decreasing in values. So these are, with three numbers, we have 6 possibilities, and the main idea is to see of these patterns without worrying about the actual numbers, just about the type of oscillation that you have, if there is an oscillation or is a trend. No, we don't care whether it's a 7 or a 70. The pattern is the same. The idea is to calculate the probabilities of occurrence. Of course, if we consider patterns with longer data points, we have more probabilities, more possibilities. We need very long data sets to calculate how often this pattern occurs, how often this one occurs, and so on. So this is a problem if you don't have a lot of data, which is the usual situation in neuronal recordings, for instance. Not with lasers. With lasers, we can record as much as the oscilloscope allows, but in experimental unstable conditions allows. But in experiments, sometimes we need to limit ourselves to consider not 3 or 4 maximum, because then we have too many possibilities and it's not possible to estimate these probabilities. No, we don't have enough data. Okay, we can solve this problem if we want to look at longer patterns. We don't need to consider consecutive data points. We can skip, for instance, 9 points and analyze the patterns formed by these red dots. No? That's a possibility. We have used this possibility in particular for climate data because the results that you obtain when you compare, for instance, assuming that the data is monthly sampled, that means filtering out the weather, the noise, just take a moving window and average data points in a month's window. Well, the results you obtain by constructing patterns with consecutive months completely differ if you consider consecutive years. So, for instance, the temperature in some location in different years, the same months. So this is interesting because it allows to select the time scale of the analysis. Sorry, I go back. It's one of technique that allows you to, for instance, instead of calculating, I don't know, the histogram of values, putting together all the values, this allows to select what time scale you want to analyze. A short time scale, consecutive values. Longer time scales, you can take values sampled with different time lag. Okay, so eventually the idea is that you calculate some probabilities. Here I have labeled A, B, C, and D, F, and before it was 1, 2, 3, and 4, and sometimes I put 1, 2, 3. The labels of the patterns sometimes are different, but they mean the same thing. So these are 6 patterns that just indicate the order of the values, how they occur in the time series. And now, question is what to do with this. And as I said yesterday, these are features. We can use different techniques to analyze the set of features. For instance, we can analyze its probabilities, the values. And we can also compute, do some sort of dimensionality reduction and compute some entropy or complexity, and Osvaldo will talk about this, the entropy complexity plane and so on. Okay, so basically point B is left for the next class. Now, main question we need to think of is, are they equally probable? So if I have an histogram like this, is this consistent with uniform probabilities? Maybe it's just that I don't have enough data, but if I had more data, I will have that a distribution that is uniform. All the patterns occur with the same probability. If that's the case, then it will mean that there is no temporal structure in the data. All the patterns are equally probable. It's highly likely that the data is generated by a fully random process. No? If your spikes or whatever analysis data you're analyzing, if you see that the patterns are more or less the same, occur with the same probability, then it's possible that the data is just generated by a random process. Now, how can we say this? We use a null hypothesis, which means all the patterns are equally probable. The number of possible patterns is d factorial. And then analyze. Is enough that one probability is outside the interval of confidence to reject this null hypothesis. So that's the criterion. If all the probabilities are within some interval of values, okay, they won't be exactly this value because this is in the thermodynamic limit of infinite data, right? So if I have a certain limited number of ordinal patterns, n, let's say, then I can define an interval of confidence interval. And if all the probabilities are inside this interval, we can say, can we say something? Not really. But if one of them, at least one of them, is outside, then we can reject the null hypothesis with a given level of confidence that depends on the interval that I'm considering. Okay? So it's enough that one is outside my interval to reject. So I am identifying signs of non-complete randomness in the data, or otherwise I cannot reject the null hypothesis. I cannot say that this is not true. Okay. So let me show you an example of application of this technique. And I'm going to go back to the logistic map that I presented to you the previous class in a parameter region. This R is almost 4. I didn't put 4 because, well, for some numerical reasons, but it's very chaotic, the time series, okay? And if I show you a detail of the time series, this is what you see. Somewhere here, you see this type of behavior. If I compute the probabilities of these ordinal patterns, now labeled from 1 to 6, we see that number 6 doesn't exist. Now, this is peculiar, because number 6 indicates 3 consecutive decreasing values. Now, if we go again to the detail that I show you, you can clearly see that when it goes down directly. It doesn't do a step. While when it goes up, it can do a step here, it can do a step here. So this pattern exists, but the other pattern does not exist. Now, this is curious. It's a new information about the logistic map. Probably you didn't know. Okay? I mean, you know the bifurcation diagram is very well known, but now you have realized that whenever it goes down, you can anticipate that the next value is going to go up. This is some information that gives you some possibility of predicting something. You can, if you have your time series and you get up to here, or here, for instance, you know that the next one is going to go up because it cannot go down. So basically, it's
sorry, Cristina, can I ask you a question?
yeah.
So does it has to be like with the sampling frequency? I imagine that if you, this is a numeric experiment, but let's say that you can adjust the sampling frequency, because what I'm seeing of what you are saying is that the going down is much more abrupt than going up.
Exactly.
So if we change the sampling frequency, probably we will have to extend to 4 points, or I don't know, 6.
Because this is a map.
So my question is this an interplay between, so the fact that you are just taking 3 points is because it's an interplay with the sampling frequency. So what I'm saying is that if you get a higher sampling frequency, probably you will have to get more points, let's say 5 or 6 points to get to that.
Changing the sampling frequency is this case, no? You can sample very with detail or not. In this case, clearly, if you change the sampling frequency, this pattern will occur. Actually, if you use a very small sampling time, the most common patterns are the ones that go up and down. It's trivial. You will have a trivial result, right? It's useless, essentially. You need to choose your sampling to filter out these going up and going down patterns that will be very frequent if you analyze very consecutive data points. So yes, in the case of the logistic map, there is no sampling time because the sampling time is 1. Okay? So this is a result that is actually a characteristic of the period doubling dynamics. The period doubling dynamics is like that. It goes, at least for the logistic map, and I will show you that it's robust with respect to the value of the parameter. This doesn't occur just for this particular value of R. But if you compute the histogram of values, the information provided is completely different. The information provided is that the histogram of values, the most probable values are down here or up here, and in the middle, they're not so probable. So basically, here we have a message that the information you obtain is complementary. You have to try both. You have to try methods that take into account the actual values and methods that take into account how these values occur in the sequence of data points. Okay? All right, so this is the logistic map bifurcation diagram. And now the question is, how can we do a bifurcation diagram like this, but in an ordinal sense? So the idea is to, instead of plotting the values, plotting these probabilities. So here are the values, and here we have 6 probabilities, and that's what we do. If we plot 6 probabilities as a function of the parameter R, we see that there are changes in behavior. Okay, for instance, first of all, we see that this pattern 210 is always forbidden. And we also see that the trend going up is forbidden up to a certain point, and then it appears. Actually, this point is coincident with the other point that I mentioned before of the merging of this. Okay? More or less when this merges is when this type of dynamics, 3 points going up, occur. Okay? So this is a technique to detect changes, dynamical changes in data, which not necessarily occur because of bifurcations. Okay?
Oh, yeah, thank you. Can you identify in these diagrams the points where you're going to have the drops?
The bifurcation points, the Lyapunov exponents.
That will be on with the Lyapunov. Okay.
The Lyapunov exponents will give you a different information. The Lyapunov exponents take into account the amplitude of the values, while here it takes into account the order in which they appear. So this is just a different type of information.
Cristina, can I ask you a question before going? It's just a follow-up of Anna's question. Because how do you know what the sampling frequency is the right one? Do you have any kind of quantitative measure for it?
Very excellent question, excellent question. There is no such thing as the right one. You need to try different ones because complex systems are multi-scale in general. So you need to, whatever sampling you choose, you'll get some results, okay? So that's the problem. It's like there's no right one between months and year. You have some, you can uncover some phenomena that acts in yearly scale, and then you can uncover phenomena that acts in a monthly scale or seasonal scale. Yeah. So you need to try to not to use redundant data. But if you can sample like this, or you can take longer sampling times.
I have a follow-up to that question. How about filters, filtering?
Features.
Filters. Yeah. Filtering the data. If you have a sampling rate like 100 hertz, when you filter your data, filtering.
Filter the data. Okay, yes, if you filter the data, you might have different patterns.
Okay.
Very clearly, yeah. In a way, it's the same as considering monthly data or yearly data. It's different data set.
But it's trial and error, right? There's no...
It's worth to try everything.
Yeah, okay.
Okay, so this method has been used in particular to test if a model is good for some data. And that's an example I will give you. To fit the parameters, assuming you know the parameters, you know the model, you can find parameters that give you the same distribution of probabilities, or as similar as possible. And also to classify different type of data without using any model, just based on the probabilities. So let me give you an example first on ECG signals. So this is kind of the raw data. From here, you can define spikes. You can then define the time between spikes, and then consider patterns using the time between the spikes. No, if two spikes are very close. Okay, so forget about the amplitude of the spikes. What we will analyze is the inter-bit times. They seem very periodic, but they are actually not, and there are small differences. Okay? So basically, in this paper, 10 years ago, they found that by plotting one probability versus another, I will now explain what these numbers mean, but basically, it's a probability of a pattern versus a probability of another pattern, you can separate a group, these control people, with people that have cardiac disease. Okay, so basically you can separate these two groups, just analyzing the raw data, no? Because basically this type of analysis is unaffected by extreme or outliers, okay, because they only affect one pattern, because outlier numbers, because we don't take into account the actual values, if there are error in the values, this very robust this technique. And here the probabilities are normalized to the smallest and largest value. So, of course, if you add all the probabilities, should give you 1, but you can always normalize to the larger value in the data set. That's why this goes up to 1, and this goes up to 1. So basically, this is an example of thinking. These are my features, as we explained yesterday. These are my features, and I find two features that if I plot one versus the other, it allows me to separate my data into groups. Okay? Now, we can use this for trying to uncover similarities between lasers and neurons. This is my time series recorded with in the laser lab, and this is an intracellular recordings. You can see here, for instance, a pattern that if we have an interval small, then a longer, and then a long, but not as long. And here we have a frequency increasing longer intervals, and here we have this pattern. So we can contrast experiments and simulations and see if the probability of the patterns are more or less similar or not. That's the idea. And this is the simulation done by Pepe Aparicio Reynoso a few years ago. We have the neuron model. We increase, because in the Figus-Nagumo model, without any external input, the spikes are induced by noise. If they are just induced by noise, we expect all the probabilities to be consistent with the equal probabilities, okay? So it's just spikes are induced by noise, and then all the probabilities should be within my gray region of confidence. All the probabilities are the same, essentially. But if I increase, if I put an input signal to the neuron model, sinusoidal forcing, and I increase the forcing, I see that I didn't put these values, but you can go to the paper and look at the numbers. Two probabilities are outside this gray region. So basically what we see is that these two patterns, which are the trend patterns, become less probable, and the other ones become more probable. And we can do the same with the laser. We can force the laser. We can apply a sinusoidal signal to the current of the laser. And if the forcing signal is large enough, we have the same behavior. We see that these two probabilities become less frequent, and these other four become more frequent, more probable. And more importantly, we see the same order. We see also that there are pairs of probabilities which have the same, more or less the same values, clusters. Okay, so we see that the values might be different. The probability values in the vertical axis are not the same, but the structure of the data and the structure of these probabilities are the same. So basically, we can say that if we have a time series generated with a large forcing, and we calculate these probabilities, and we see that they are not in here, we can see that the laser or the neuron are perceiving some input and are encoding this input in the occurrence of these patterns. It's a way of encoding information. And they have the same two groups of patterns with the same probabilities. Okay, so this is interesting, but might be relevant, maybe yes, maybe not. Nowadays, we are worried because these artificial intelligence systems, high-performance computing systems, consume huge amount of power. This is a problem in the context of climate change. This data center of the European Center of Medium, where it consumes the same as a town, okay? So if we can somehow mimic how the human brain processes information with only 19 watts, then it'll be a huge development, no? So that's the point. There's a lot of people working on developing photonic neurons. These photonic neurons not only will be faster, orders of magnitude faster, but will also be much more efficient. Okay, much less energy consumption. So there is a field that is called neuromorphic photonics that I think maybe you talk about this too, okay? So there's lots of work going on trying to develop artificial neurons that can be faster, but more importantly, less energy consumption. And basically, the point is that when you go to use lasers with low power, what you need is to have a code to encode information that is noise robust. So the difference between why the brain is so efficient basically is on neural coding. And that's what is not being implemented at the moment. Artificial intelligence systems operate in a completely different way. They don't use neural coding, in particular because neural coding is not well understood. Nobody knows. So before moving to this, I tell you that the laser has this type of time series which look similar to neurons. The main challenge is to understand the neural code, to understand how neurons encode information, how use spikes to encode information, and maybe also use noise to encode a small amplitude information, how neurons are able to perceive very, very weak signals. So basically, the idea is to understand how this how this signal in the Figu-Nagumo model can be encoded. And this is what we did. I showed you before some results. These are similar for some other amplitude or some other level of noise. It all depends. See, for instance, the probability depends on the period of the signal and also depend on the amplitude, as I showed you before. Which probabilities become more probable depend on the period and on the level of noise. There's a lot of parameters here to tune, but one can wonder, well, maybe this is just encoded in the rate. Why not? The spike rate can encode, for instance, the period of the signal, right? It's possible. It's possible, but it doesn't happen. Because if you calculate the inter-spike interval, the average, so the inverse of the spike rate, you see that there is a wide range of locking. The system is locked. It's locked to, so independent of the period, the spikes are locked. The inter-spike interval is almost the same. So you cannot encode the period in the rate. But it could be encoded in these probabilities because the probabilities differ. They have different values depending on the period. So if we can now go back and say, from these probabilities, can we recover the period? Suppose we don't know the period. So that's what's going on in our lab at the moment. Now, other issue is that this type of coding mechanism will be very slow if you use it at the level of a single neuron. Basically, because you have to calculate these probabilities. The dynamics is very noisy, they are very similar, so you really need long time series, long sequences of spikes. But however, you can use 1,000,000 neurons, or 1,000 neurons, or 100. And if you do this, then it will be fast, because you only need 2, 3, 4, or 5 spikes per neuron to then calculate the probabilities over the whole ensemble of neurons and see if you can encode the information in the probabilities of the ordinal patterns. So basically, calculating the probabilities just with a long spike sequence is slow, but with a short spike sequence, but with many neurons, can be very fast. So a student, Cristina Masoller, did her PhD thesis on this and considered an ensemble of neurons, basically coupled with some random architecture here, and applied some weak signal here, sub-threshold by itself cannot produce any spikes, and we found that effectively it's possible, again, increasing the amplitude to see that some patterns become more probable, others less probable. These, again, depend on the modulation period. You have some noise resonance with some level of, if I put too much noise, of course, we expect that all these probabilities will again become the same, okay? If I don't have noise, there are no probabilities because there are no spikes, but in the middle, there are some noise that helps to encode, and you don't need too many neurons. Increasing the ensemble size does not change too much after a while. You need maybe 10 or 20, and then that's the same. And again, the probabilities of connections, very few connections is enough to encode, to have these probabilities different from the uniform. And the coupling strength doesn't make a lot of difference again. So now the question is, how can we recover from the probabilities the values of the period, the amplitude of the signal, and if we can use this for a periodic signal, signals of other forms, etc. Okay, so can we compare lasers and neurons? These are simulations and experimental work done by Jordi Tian and Carlos Quintero. And again, this is a picture of a lab. And if we use the spike rate, so basically what we are plotting here is the inverse of the inter-spike interval in color code. We see with a sinusoidal perturbation or a pulsed signal, we see that there are regions of a high spike rate. There are regions in which there are very few spikes, and more or less they are comparable. There's some problem here that this pulse signal generates something that the sinusoidal signal does not. But if we go to compare with the neural model, we see some similarities with the Figu-Nagomo model. We see, especially with pulsed signal, we see the same. But with sinusoidal, we find differences. We find this third region, which is not here. And if we go beyond the spike rate analysis, and we look at temporal code, the ordinal patterns, we find very, very difference, a lot of difference. So this is in color code, which pattern is the most expressed. So for instance, in the yellow region, this pair, which always take the same value more or less, we add them up, is the most probable pattern. But in the Figu-Nagomo model, you can see the structure is completely different. Meaning, we find similarities with the spike rate, but no similarities in the temporal structure of the data. So we need to work far harder to find conditions in which our lasers really mimic or really reproduce the behavior of neurons, both at the level of the spike rate and at the level of the temporal correlations. Software. You can use, this is available on the web page, and this is, I will give you an exercise for hands-on if somebody wants to do it. You can press this link, and in the appendix, you have the software. I have it here in MATLAB, but it's also available in Python. The code is available in this paper in the appendix, okay? And this is available in the web page, so you can download it from the web page of the school. Okay. Now, what does it mean? What do you need to give? How this algorithm works? It's quite simple, but you need to play a little bit with it to become familiar. You give the time series, TS, but you don't give all the time series. You just give the window from where you want the pattern. So you call this recurrently. Okay. You cannot send all the data and it gives you all the patterns. It doesn't work like that. You send a window or a set of values, and it returns the pattern. And then there is the word length. Word length is 4, for instance. And you can also choose the lag, 3. So skip 2 points. So you have to give, if you use these settings, 4 and 3, you have to give 11 data points, no? So 4, and 4, and 4, okay? So you have to give exactly these data points, and it will construct, it will return the index. This is what it returns, is the index 3, which corresponds to this pattern in this list, okay? So basically, this gives you the ordinal pattern, and then you apply this through your data. You get the sequence of ordinal patterns from where you can calculate these probabilities. And the hands-on exercise is exactly to analyze the logistic map. I suggest, before trying with your own data, that you don't know what to expect, make sure that this works for you and you reproduce what you are expected to obtain. So You first try the pattern, for example, with numbers like this, which will return the index 3. This is pattern number 3. Then try longer words with lags, okay, to make sure that you understand how it works. And then you can generate a time series with the logistic map, calculate the ordinal probabilities, and then change the control parameter to reproduce the ordinal bifurcation diagram. So this is the exercise, and a possible exercise for you if you are interested in doing some hands-on exercise with programming. The language, the algorithm is available both in MATLAB and in Python. Sorry? You can superpose or not superpose, okay? If you want, you can superpose, but then if you want to analyze what they call transition probabilities, which is the probability of a pattern occurring after another, you have to have non-superposing cases, okay? But if you just all calculate probabilities, then it's not a problem to have one pattern for each data point. 16 points. 6 points. It's too small. Well, you can have, you should calculate probabilities over the population of patterns, or over the population of patients. But then you cannot distinguish patients, right? Mm. No, you need data here. You need data. So basically, the plan is to simulate this. This is easy. Start with an initial condition, get the next value, and so on. So you have a time series, and then apply the algorithm to calculate these probabilities. Make sure that the pattern 210 does not occur, and then change the control parameter and do the bifurcation point, the bifurcation diagram. Okay, I have not enough time to show what I wanted to do, but I can finish at least with the application of this. We have done an application of the analysis of EEG recordings. This is one subject with 16 records from different areas of the brain at a certain point, in this case, 2 minutes. The patient opened the eyes. We see a transition here. We see that the time series doesn't look the same, but we want to distinguish, so we can use this technique. We have used 2 data sets. One is free from Physionet, the other one is not free. And we calculated this permutation entropy. So that's the only equation I'm going to put. From these probabilities, we calculate the entropy with the usual Shannon entropy. And Carlos Quintero, in collaboration with other people in our group, did this analysis and found that the entropy, has this, is lower, essentially. This is the data set of 1 minute and 1 minute, and the data set, the entropy is lower, but doesn't allow complete separation. So a few months ago, we discussed this with Bruno. Bruno explained this. So Bruno improved this by computing this entropy in a different way. The way was, instead of computing the ordinal patterns in time, computing the ordinal patterns in space. So using the values of the electrodes at a given time. Okay, so we have 16 electrons, or 74 electrons, so that give me a certain number of patterns. And then use the different times to compute entropies as time changes. So basically, this is how as a function of time, the time series with the eyes open is up here, the eyes closed is here. And when we plot with error bars, we see a better separation than in this case. So in this case, in each time series, I compute the ordinal patterns, okay? And in this case, is at each time I compute ordinal patterns. Yeah, if you shuffle the electrons, you cannot distinguish. So there is information in the spatial pattern organization of the electrons. The ordering is related to the ordering in the brain, yeah. But it doesn't mean that it's the best ordering. We didn't try all Bruno didn't try all the probabilities, not possible. Okay, so we also try this for vegetation transitions. And here what we have is images, remote images in a place, in a space. This is a space, and there is no vegetation here, and there's a lot of vegetation here. So we have the average tree cover, more or less, and we have the rainfall. And we see that here, in a distance of 120 kilometers, we have no rain or a lot of rain. And if you plot one versus the other, there is some transition like this. And what I want to briefly show you is that this technique also allows to anticipate this transition, to anticipate that the vegetation is going to be extinguished as we cross this kind of border, okay? So basically, we computed the entropy, also the permutation entropy, as a function of the rainfall, and we see a point in the control parameter in which the entropy clearly goes down. The usual correlation, the usual indicator, which is when you approach a bifurcation, there is a critical slowing down, and then because of critical slowing down, there is a spatial correlations. It doesn't display this monotonic behavior. So basically, this is another indicator. This entropy can be used as an indicator of approaching bifurcation, because the spatial entropy, permutation entropy, can be calculated as a function of a space, and we see how, as a function of a space or as a function of rain, because it's more or less kind of linear here, more or less. We could put a space here too. This goes up, and then on the other side, it doesn't change too much, but also goes up approaching this bifurcation point. So I'm going to finish here, and I will leave this for my class tomorrow. Yeah. Questions?
Thank you, Cristina. Thank you very much. I have a question, a general question. Can you go back? Let me see. I'm very curious about something. If you can go back the slides. Wait. One more. This one. In that time series, if I add white noise to the data.
No.
If I do that, if I add white noise, if I add some artifacts, do I destroy completely the results, or it is resilient to noise?
I don't know.
Okay.
Good question, but I don't know. I don't know if Bruno,
someone can
you
try.
didn't test white noise, no? Here, for this. The data is noisy enough, yes.
It's noisy, but is the analysis resilient to noise? I mean, have you tested? Maybe if you do some averaging, or if you do some denoising, let's say, with a Fourier transform or whatever, can you clean up the data somehow?
He did try. I can answer that. He did try filtering out using the frequency wave, the wave that is more pronounced for eyes open and eyes closed. But he didn't add noise. And I think there is a very good question, because it's possible that sometimes adding noise improves the differentiation. Because it's possible that one state is more robust, the other is less robust, and you can separate more. It is maybe a good idea to add a little bit of noise and plot the separation versus the noise level. Maybe it just goes down, and when you put a lot of noise, you cannot separate, but maybe you can have a curve like I show you, with some resonance, with some level of noise, the probabilities become more different from uniformity.
Okay, thank you.
Yeah. Oh, he will discuss. Excellent. Additive noise, yeah.
You said that for codify your symbolic analysis, you used the time between the spikes. But is there some importance on the magnitude of these spikes?
In theory, no. If it's excitable system, the amplitude is the same with respect to the perturbation. The amplitude doesn't encode. The amplitude of the spike does not encode information of the perturbation. Because if I perturb a little bigger, the spike is the same. It doesn't encode information on the timing, because if I have two very close perturbations, the second one is not, doesn't do anything. So the timing is important, but the amplitude, if the system is excitable in an ideal condition, no?
Still about this result, what S1, S2, S3 means? The blue, red, and green rectangle.
This here?
No.
This?
Yes.
1, 2, 3.
No. The S1, S2, and S3.
Yeah, these are two free letters that we use to encode a symbol. So the value here, and the value here are used, and then I obtain a symbol from these three numbers. And here
okay.
we get that, and the last pattern, the last one, is obtained from these three.
Okay. But
so it's just, what we do is transform a time series of real values. These are real values. I think they are all positive, maybe, I don't know, into a number of integer values. So these first three, give me a number S1. S1 is a number between 1 and 6, if I'm using 3. If I use 4, it'd be a number between 1 and 24. Then the next values, 2, 3, and 4, give me another symbol. This is symbol 2. And then so on. These 3 values give me symbol 3, and so on. So I have 64 symbols. 64 symbols, and I have to compute 6 probabilities and borderline, because you need to have at least 10 values per probability, right? So 64 symbols to distribute in 6 boxes is borderline, but it's still more than 10 data points per bin, which is what you have to use if you want to compute probabilities. 10 data points per bin on average.
Okay, but I'm still confused by the order, because you use 1, 2, and 3 to make the symbol S1.
The color is the blue gives me the symbol blue.
No.
The red gives the symbol red.
I know, but why the electrodes are in order? I mean, 1, 2, 3, 4. Because there's an overlap, and you could shuffle, for example, the electrodes.
Yeah.
So
Yeah. Let's see that. Let's put it like this. If you have the electrodes in the brain, you have like this, no? Something like this. Let's say. So these are, if we label like this, 1, 2, 3, 4, 5, 6, see it? These ones give me one symbol. Yeah. Okay? Then these ones give me another symbol. Then these ones, okay? Yeah. So that's what represents the one. They are aligned in a line, but they actually have a 2-dimensional representation.
Okay.
And the way Bruno did it is just following the index, 1 to 64. Okay. But it's possible, and he actually found a better ordering, in which you probably cut here and you define patterns like this, and you can even improve this. I mean, straightforward was to just follow the numbers. But if you do it in a more sophisticated way, it's even better, and you can even separate better the two states.
Yeah, makes sense. I think there is probably an optimal separation of the electrodes that gives the better result. Yeah.
Absolutely. And the next step, natural step, would be to mix the two approaches. So to define patterns that are spatio-temporal. So they have some free values, maybe 2 in a space and 1 in the next time, for instance, things like that.
Yeah,
There is room for improving. This is interesting because you can detect, we couldn't detect at the level of the individual patients or subjects, no? It's not possible for just one time series of one subject. We have not been able so far to find a way to just, from that one, clearly separate 2 values.
Okay, interesting, thank you.
Thank you. Do you consider evidence interregional differences?
I'm sorry. Can you repeat?
Do you consider distinguish interregional differences? Consider interhemispheric or brain regions pattern?
I think it's in the same direction. If we consider brain regions patterns.
But I'm thinking in interhemispheric because...
Yes, I fully agree with you, yes. It's possible just to not mix the 2 hemispheres, or it's possible to mix them. And it's also, in a previous work in this one, we found regions which were more informative. So it's possible to do this analysis only considering those regions. When you calculate the probabilities in these regions, separate better. So not all regions are equally informative. And using this 1 to 64 to find the patterns is just straightforward, but maybe it's possible to find better ways to do this, for sure. And it's also a technique that you can use for separating other situations. No? In PhysioNet, you have data alcoholic, non-alcoholic patients, for instance, and you have data of people with epilepsy versus people with healthy subjects. So this technique can be used for trying to differentiate different types of conditions. One data set, yes, is from PhysioNet. The other one, no. The other one is from a company in Zaragoza, but it's possible to ask permission. In principle, it's possible.
Any other questions? Okay, so let's thank Cristina again. And now we have a coffee break, and then we continue.